Okay, so what's shrinkage? Shrinkage is something that DESeq2 does, and DESeq2 is better than DESeq1, just for your information. Anyway, so when you do some DESeq, there's normally a normalization step, but shrinking comes after the normalization step of DESeq2. So, like, shrinking is a thing we do, which improves the accuracy of when we do DESeq2. So there are two factors to decide whether we want to do some shrinking or not. So the first, let me just show you what shrinking does to data. So here we have an MA plot with all the genes compared to the mean of how many counts map to the RNA, to the transcript. And what shrinking does is it basically shrinks this part it shrinks it down. So it shrinks the log fold change from really high to low. But so that's just roughly what it looks like. I'll explain some more later. So there are two factors that we use to decide if we want to do some shrinking. The first thing is low gene. Okay, we'll go them in order. First thing is high dispersion value. So over here in this diagram, you can see that we have green, like genes in green, and then genes in purple. And both their means are pretty similar, but the difference is purple has really high dispersion values, so it's really high variance, whereas green doesn't. So basically, when you do some shrinking with DESeq2, what happens with green is, so you have this solid green line over here, and that's the unshrunk values. But then when you shrink them, it's the dotted line over here, and there's hardly any change. But for genes with high dispersion values, like so much variability, the unshrunk log 2 fold change, so the unshrunk one is like higher, but after shrinking it goes lower. So shrinking makes genes with high dispersion values shrink towards 0. So if it was negative as well, it would shrink towards 0. So it's like a more conservative way of it's being more conservative with the log 2 fold change values when you do some when you want to see which genes are differentially expressed or not. So that's the first thing. And the second factor we use to decide if we want to do some shrinking is low gene counts. So that was like shown here. So basically all the genes over here have higher gene counts. And so there's not gene counts. Like higher counts mapping to the gene. So all these guys over here, shrinking doesn't like affect it because it has really high like counts. But at the lower end of the spectrum, all these guys are shrunk towards 0, the log fold change of 0. That should be log 2 fold change, but anyway, you get the idea. So yeah, that's shrinking. And then I can explain this figure here. So basically, each black dot here is the gene-wise maximum likelihood estimates. So yeah, maximum likelihood estimates. So these are individual genes, and so they're the gene-wise maximum likelihood estimates of the dispersion. And then what you have in red here is you just fit a red curve to the black dots. So it's just to capture like an overall trend for how dispersion and the mean of the counts. Yeah, it's just to capture the trend of these two. So of these two axes. Then you use this red curve to for like a second estimation round, which is used to shrink the black dots or the individual genes. So that's roughly what this figure is showing. Yeah. So if you wanna if you wanna read more, then I'll post the references to a paper that describes this really well in the information box of this video. Okay, thanks for watching. That's roughly what shrinkage is. Yeah.